Good morning, students. Now coming to post fertilization structures and events. You know, all the structures formed after fertilization they are considered as post fertilization structures. These post fertilization structures they includes endosperm. They includes endosperm, okay, and zygote and formation of embryo, that is called embryogenesis. Formation of embryo, that is embryogenesis. At the same time, formation of fruit from the ovary. Formation of fruit. So ovary develops into a fruit after fertilization. Similarly, ovules present inside the ovary, they develop into seeds. So ovary develops into a fruit, ovules develop into the seeds, and fruit and seeds also post fertilization structures. And remaining flower parts means calyx, okay, and corolla and stamens, okay, and style and stigma, and all these floral parts completely degenerate. These floral parts they are completely degenerate. They degenerate after fertilization. These parts of the flower they are completely degenerate, fall off and degenerates, and endosperm, zygote, embryo, genesis, and formation of fruit, formation of seeds from the ovules, all of them they are considered as post fertilization structures. First, coming to the first post fertilization event, that is endosperm. First, coming to endosperm. Students, this endosperm, formation of endosperm, it proceeds embryogenesis. Formation of endosperm, it proceeds embryogenesis, embryo development. Why embryological development is dependent on endosperm? Endosperm, it is the food for embryo. It is the food for embryo. So endosperm is nothing but the tissue which provide nutrients to the developing embryo. So endosperm it provides nutrients to embryo. Because of this reason, this is considered as the food for embryo. So actually, endosperm it is developed from primary endosperm nucleus. This is triploid. Okay. Primary endosperm nucleus is the product of triple fusion, where second male gamete is fused with the secondary nucleus, leading to the formation of primary endosperm nucleus. This is a triploid. Okay. This primary endosperm nucleus, this is the initial cell for the development of embryo. What is the initial cell for the development of endosperm? Okay, development of endosperm. What is the initial cell for development of endosperm? Primary endosperm nucleus is a primary cell for the development of endosperm. Okay. For example, this is a primary endosperm nucleus, pen, and this is a triploid. The nucleus of primary endosperm nucleus, it undergoes free nuclear division. Free nuclear division means the continuous nuclear division without a cell division. That is called free nuclear division. So, endosperm mean primary endosperm nucleus, it divides by mitosis, leading to the formation of many nuclei. Many nuclei. This is called free nuclear division. Primary endosperm nucleus divides by mitosis, leading to the formation of multinucleated primary endosperm nucleus. Now, this stage of endosperm, this is called nuclear endosperm. Nuclear endosperm. This is called nuclear endosperm. This nuclear endosperm, it is also considered as liquid endosperm. This is called liquid endosperm. In this way, free nuclear division leads to the formation of nuclear endosperm. Now this nuclear endosperm, this is also called liquid endosperm. One of the best example for this nuclear endosperm, the water, coconut water. Okay, coconut water. Tender coconut water is nothing but nuclear endosperm. So this liquid endosperm is represented by thousands of nuclei. This is one of the example. Now, in subsequently, all these nuclei, they are followed by cellularization, resulting in the formation of cellular endosperm like this. Cellular endosperm is formed by the process of cellularization. So, this is a cellular endosperm. This is a cellular endosperm. So, this cellular endosperm is also called solid endosperm. This is called solid endosperm. So, as a result of the cellularization of nuclear endosperm, it leads to the formation of cellular endosperm. This is also called solid endosperm. One of the best example for this solid endosperm, white kernel of coconut. White kernel. Okay, white kernel of coconut. White kernel of coconut means copra. Copra is one of the best example. So, nuclear endosperm, it is converted into cellular endosperm due to cellularization. And the cell walls and the cells are formed from this nuclear endosperm, leading to the formation of solid endosperm. This is a best example, white kernel of coconut, that is copra. In this way, primary endosperm nucleus first it develops into nuclear endosperm by free nuclear division. Then this nuclear endosperm is converted into cellular endosperm due to cellularization, resulting in the formation of endosperm. This is endosperm. So this endosperm, this is very rich in nutrients. It is very rich in nutrients. Very important. Endosperm is very rich in nutrients. So it provides food materials to developing embryo. It provides food materials to developing embryo. But in some members, very important. Students, in some members, this endosperm is completely consumed by developing embryo. In some seeds, embryo it completely develops the endosperm. Means endosperm is completely utilized by the developing embryo, so that such seeds have no endosperm. Why these seeds have no endosperm? Because embryo it consumes the total endosperm, so that there is no the endosperm. Such seeds are called non-endospermic seeds. Very very important. Non-endospermic seeds. Okay, these non-endospermic seeds they are also considered as exalbuminous seeds. Exalbuminous seeds. They are also called exalbuminous seeds. These exalbuminous seeds have no endosperm because endosperm is totally consumed by developing embryo. So that seeds are non-endospermic and also called exalbuminous seeds. Some of the best examples for these exalbuminous seeds, Fabaceae members. Fabaceae members. What are some examples for these Fabaceae members? One is pea and other one is bean. They are best examples. So, members of Fabaceae like pea and bean, endosperm is completely absent inside the seeds. Why endosperm is completely absent? Totally this endosperm is consumed by developing embryo. So that endosperm is completely absent. Such seeds are called non-endospermic seeds or exalbuminous seeds. Example Fabaceae members like pea and pea. Okay. Now coming to another point. It says, in some seeds, endosperm is not completely consumed by developing embryo. So, embryo it consumes a part of endosperm. Embryo it consumes some amount of endosperm and the remaining endosperm is left out inside the seed. Okay, such seeds with endosperm, such type of seeds they contain some amount of endosperm because endosperm is not completely consumed by developing embryo. Such seeds are called endospermic seeds. Endospermic seeds. Okay, endospermic seeds, and these endospermic seeds are also called albuminous seeds. Albuminous seeds. Endospermic seeds are also called albuminous seeds. Some of the examples for these albuminous seeds, coconut is one of the example, and castor is also one of the example. So, coconut water is considered as a nuclear endosperm, and coconut cellular endosperm is a solid endosperm. So, these type of seeds they contain some amount of endosperm. Seeds with the endosperm are called endospermic seeds, also called albuminous seeds. Example, coconut and castor. Okay, now coming to the most important points here. One of the assertion and reasoning question. Seeds of pea and bean, they are non-endospermic seeds. This is assertion. Is right. Seeds of pea and bean, they are non-endospermic seeds. Reason. These seeds contain endosperm. This is right. Correct explanation. Seeds of pea and bean, endosperm is not formed. In seeds of pea and bean, endosperm is not formed. This is wrong. Seeds in the seeds of pea and bean, embryo does not consume the total endosperm. Seeds in seeds of pea and bean, embryo it consumes the total endosperm. This is right and correct explanation. Why? Because initially endosperm is formed inside the seeds, but it was completely consumed by the developing embryo, so that seeds are without endosperm. They are considered as non-endosperm. and exogenous seeds. Now coming on the very important point, students, in case of Orchidaceae, family Orchidaceae, okay, and family Podostemeraceae. Podostemeraceae. In these two types of family members, endosperm is not found. Endosperm not found. So endosperm is not found. For example, Orchidaceae. This is one of the monocotyledonous family. Members of this Orchidaceae, endosperm is not found. Endosperm is not found. So that seeds are the non-endospermic. Non-endospermic. Seeds are the non-endospermic. Students, seeds of Fabaceae and seeds of Orchidaceae, they are non-endospermic seeds. Both are non-endospermic seeds. But what is the reason? Reason is different. Here, endosperm is initially formed inside the seeds, but it was completely consumed by developing embryo. So that endosperm is absent in the seeds of Fabaceae members like B and B. But what about the reason for the absence of endosperm in the seeds of Orchidaceae? Here also endosperm is absent in seeds, so that seeds are non-endospermic. But reason is different. Here, initially endosperm is not formed inside the seeds. Here, formed inside the seeds but completely consumed by developing embryo. So that seeds are non-endospermic. Here, initially endosperm is not formed inside the seeds. So endosperm, these are the non-endospermic seeds. Very important. So endosperm, it is nothing but nutritive tissue for developing embryo. And initial cell for this endosperm, primary endosperm nucleus. Now coming to another important point. In case of angiosperms, endosperm is 3N. Endosperm, ploidy of endosperm is 3N. Triploid. But what about in case of gymnosperms? In case of gymnosperms, endosperm is haploid. Endosperm is haploid. Why endosperm is haploid in gymnosperms? Female gametophytic tissue is directly serves as endosperm. Female gametophytic tissue is haploid. Now this tissue, it is directly acts as endosperm. So that endosperm in gymnosperms is haploid. But in case of angiosperms, endosperm is triploid. Why it is triploid in angiosperms? This is the product of primary endosperm nucleus. It was formed by the triple fusion in case of angiosperms. Okay.